Morning everyone. Today we are going to discuss about hybridoma technology, production of monoclonal antibodies. By the way, myself, Maheshwari Palipanka, Associate Professor, Department of Pharmaceutical Biotechnology, AIM Memorial College of Arts. So today we are going to discuss about the hybridoma technology, that is for production of monoclonal antibodies. In 1975, these technology developed by Georges Fogler and Cesar Milestone. And in 1984, they shared a Nobel Prize for this discovery. They make a hybrid cell that will make a number of monoclonal antibodies against the antigen. So first coming to know what are monoclonal antibodies. Before going to discuss in depth about the production of monoclonal antibodies, first of all, we have to know what are monoclonal antibodies. Monoclonal antibodies are nothing but these are the antibodies which are working against a specific antigen that are completely produced from one single clone of plasma cell or one single clone of B cell. And that is, they are having all similar properties. They are very much resemble to one to one, so that they are called as monoclonal antibodies. So they can able to detect one single epitope or single antigenic determinant that is present on the surface of the antigen. The hybrid cell has the capacity of antibody production derived from B cells, that is splenic B cells. At the same time, it can divide continuously by the quality derived from myeloma cells. By combining the desired qualities of both the cells, the technology ensures large antibody production of single specificity. Specific hybridomas, spleen cells and myeloma cells obtain monoclonal antibodies in artificial media. This technology called as hybridoma technology. So what I'm saying is hybridoma technology is defined as the technology by which we can able to fuse both the cells, that is B cells and myeloma cells. B cells or plasma cells are producing the antibodies. They are having the property to produce antibodies, that is splenic B cells. When coming to the myeloma cells, these are immortal. So immortal nature helps us to survive the cells for longer periods. So that we are fusing the two cells, that is myeloma cells and B cells or plasma cells, so that we are producing the monoclonal and we can able to produce the monoclonal antibodies with the help of these hybrid cells. So that is the principle behind the hybridoma technology. Again, I'm repeating, the hybrid cell has the capacity of antibody production derived from B cells. So these splenic B cells we are fusing with the myeloma cells. Myeloma cells are nothing but cancerous cells which are immortal in nature. They don't die after certain time period. So the immortal nature very helpful to us to survive for longer periods. The cells can able to sustain for longer periods. So the myeloma cells and B cells we are fusing to form a hybrid cells. These hybrid cells are able to produce the monoclonal antibodies. So here we are intentionally injecting an antigen to this mouse. So after 72 hours of the injection of antigen to the mouse, this mouse spleen we are extracting after 72 hours. From that spleen, we are isolating the splenic B cells, that is B lymphocytic cells. These lymphocytic cells can able to produce the plasma cells. Actually, the plasma cells are producing the antibodies. Apart from this, we are also taking the cancerous plasma cells. These cancerous plasma cells are immortal in nature. So these two we are combining to form a hybrid cells. These hybrid cells are used for the production of monoclonal antibodies. Monoclonal antibodies are antibodies that are identical with each other because they are produced by single clone of immune cell, all clones of a single parent cell. Basically produced by white blood cells, which is called as the plasma cell. It is also used for the treatment of cancerous cells as well as antivenom. There also acts as antivenom, that is toxic to the snake poison. Procedure, immunization of uh, specific animal while while they are generating hybrid cells, hybridoma cell with the spleen cells, isolation of myeloma cells, fusion between the spleen cell and myeloma cell, selection of fat medium. These are the simple steps involved in the procedure. So first of all, we have to procure the B cells, splenic B cells that are we are isolating from the mouse after injection of the antigen. Then we are isolating the myeloma cells, and we are fusing with these two types of cells with the help of polyethylene glycol broth. Apart from this, we have to have the hat medium isolation of then in hat medium the propagation of the cells will be taking place. After proper propagation of the cells, that is proper growth of the cells which we want exactly, we have to isolate these hybridoma cells, and then we can screen for hybridoma cells. Immunization of specific animal, an antigen immunized to an animal, that is intravenously or intravenous injection. Through intravenous injection, we are injecting an antigen to the animal, that is mouse, where in spleen it activates B cells which produce the plasma cells. Plasma cells can able to produce the monoclonal antibodies. So first of all, we have to isolate the plasma cells from the spleen of the animal. Isolation of myeloma cells. Myeloma cells are cancerous cells which isolated from bone marrow. Myeloma cells are generally immortal in nature. Uh, that can able to, apart from the immortal nature, myeloma cells are actually they're having another property. That is, they can also produce their own antibodies, but actually we don't want this property. That is why we are inducing the mutations, at least two mutations to these myeloma cells, so that they will lose the nature, that is capability to produce the own antibodies. So after mutations, these myeloma cells only having they can only having the immortal nature. Now we can fuse these myeloma cells with the help of PEG polyethylene glycol broth to that B cells, splenic B cells. Fusion of spleen uh, and myeloma cells, splenic cells and myeloma cells. It requires the polyethylene glycol medium. It can also done by electrofusion. Fusion between spleen cell and myeloma cell produced 5 different types of cells. So 5 different types of cells, that is we can see in this picture. These are the splenic B cells which are having the HGPRT. Just remember this HGPRT. I can explain what is HGPRT, what is the importance of HGPRT later. So first of all, we are taking the splenic B cells. Then here we are taking the myeloma cells. These are cancerous cells and immortal in nature. And these splenic B cells and myeloma cells, these two types of cells are fused. So we are putting them, the both type of cells, in polyethylene glycol broth or medium, so that we can have the different types of cells, that is unfused splenic cells, unfused splenic B cells. These are fused splenic B cells, means fusion taken place in between the splenic B cells. Here, this is the hybrid cell in which we can have the splenic B cell and myeloma cell. So this is actually we want. These are the hybridoma cells. And apart from this unfused splenic B cells and fused splenic cell and myeloma cell, we also have the fused myeloma cells, means fusion taken place in between the myeloma cells. And these are unfused myeloma cells. So total 5 types of cells we are having. So fused plasma, fused myeloma, hybridoma, unfused plasma, and unfused myeloma. So next, after that fusion, we have to isolate the hybrid cells. How can we isolate the hybrid cells? That means with the help of hat medium, we can able to isolate. Before multiplication of antibody, it has to be synthesized. Has to be synthesized new copy of DNA, and for that requires synthesis of nucleotides. So first of all, to synthesize the new cell, first purines and pyrimidines, that is nucleic acids, has to be synthesized. Then from that, the new cell will be synthesized. So for the synthesis of purines and pyrimidines, the possible pathways are salvage pathway and de novo pathway. In first pathway, that is salvage pathway, it requires degraded part of old nucleotide to produce new nucleotide. I wanted to emphasize on these two pathways in brief. Salvage pathway means already, first of all, purines or pyrimidines means nucleotides, that is nitrogenous bases, nitrogenous base, sugar and phosphate. three things to be combined to form the nucleotide. That is, in case of purines, if you consider purines, we have to have three types of the components. First of all, nitrogenous base and sugar, then phosphate. So in Salvage pathway, already these building blocks are available. Then these are combining to each other to form the nucleotide. When coming to the de novo synthesis, only there is no ready raw materials for the synthesis of purines. Total synthesis has to be taken place. That is, has to be taking place. That is, for the nitrogenous base and phosphate and sugar, nothing is there. No one is readily available. So first of all, nitrogenous base has to be synthesized, then phosphate has to be synthesized, then ribose sugar has to be synthesized. Then these three molecules are combining with each other to form the complete nucleotide, that is our purine. So de novo synthesis is it is synthesized completely new nucleotide by small molecules, that is a sugar, amino acid, etc. So Salvage pathway it requires degraded part old nucleotide, already available old nucleotide is there. So from that, just some sort of small chemical reactions will be taking place. That is addition of the ribosyl phosphate or only ribose or phosphate moiety, we are getting the nucleotide. So this is the difference between Salvage pathway and de novo synthesis. De novo means new synthesis. This is completely new synthesis. Salvage pathway means already building blocks, small amount of nucleotides and nucleosides available, and only phosphate moiety or sugar moiety we have to add. So in hat medium, cells are not synthesized de novo synthesis due to the presence of aminopterin in hat medium, which blocks the dihydrofolate enzyme, which is necessary for this synthesis. So whenever we are discussing about the multiplication of the cells, first of all, nucleic acids has to be synthesized. Then these nucleic acids formation completed, then the new cell will be formed. For the synthesis of nucleic acids, two pathways are available. But when coming to the hybrid cells, we are now providing the hat medium to isolate the hybrid cells. So how with the help of hat medium we can able to isolate or we can able to recognize the hybrid cells means in hat medium we are having three components, that is hypoxanthine, aminopterin and thymidine. Hypoxanthine, aminopterin and thymidine, these three are must and should in hat medium. So aminopterin is the component which can able to block the de novo synthesis of purines. So because of this blockade of de novo synthesis, the only available pathway for completion of the purines synthesis is Salvage pathway. But for synthesis in Salvage pathway, it must require HGPRT enzyme. But in myeloma cells, they are lacking the HGPRT. HGPRT means hypoxanthine guanine phosphoribosyl transferase enzyme. So because of lack of HGPRT enzyme, the myeloma cells, basically they are immortal, but they cannot able to synthesize the purines. So they can't able to grow in hat medium. When coming to the B cells are having HGPRT, but they are not immortal, they are mortal. They can die after certain time period. So they grow initially in hat medium, that is splenic B cells. They can grow initially in the medium, but after certain time period, they are going to die. So we can understand here is the fused cells, only hybrid cells can able to sustain or can able to grow in the hat medium, where hypoxanthine and thymidine are also used as precursors for the synthesis of purines. Isolation of hybridoma cells. Spleen cell have HGPRT enzyme. Myeloma cell doesn't have HGPRT enzyme. So disease can able to grow in hat medium but can't able to survive for longer periods. This is unable to grow because of lack of HGPRT enzyme. And without growing, they can't able to exhibit their immortal nature. So that we can only expect the growth of these hybridoma cells among these five types of cells in hat medium. Fused myeloma and unfused myeloma cells didn't have HGPRT enzyme, so can't survive in hat medium. So finally, hybrid cells has HGPRT enzyme from spleen cells and as well as their They have the ability to multiply repeatedly as myeloma cells because of the immortal nature. So isolation of hybrid cell because is only cell which survive in hat network. Screening of the hybridoma cell, ELISA screening method, ELISA screening method, which can be done by incubating hybridoma culture in which a secondary enzyme gets conjugate and formation of colored product shows positive hybridoma cells. So with the help of ELISA or radioimmunoassay, enzyme-linked immunosorbent assay or radioimmunoassay, we can able to screen the exact hybridoma cells, that is the single cells we have to take, the hybrid cells, and these single hybrid cells we are placing into the entire plate so that we can get the monoclonal antibodies, that is the antibodies which are producing from the single cell or single clone of the B cells or hybrid cells used for multiplying the hybridoma cells in vivo and in vitro. Basically, hybridoma cells are nothing but fused cells of the B cells and myeloma cells. But when coming to the monoclonal antibodies and polyclonal antibodies, monoclonal means these are coming produced from the one single clone of the B cells. When coming to the myeloma cells, when coming to the polyclonal antibodies, polyclonal means these are produced from different multiple number of cells, multiple number of B cells or plasma cells. So what is the difference? Another differences are also there. Actually, monoclonal antibodies can able to detect a single epitope or single antigenic determinant which is present on the surface of the antigen. When coming to the polyclonal antibodies, they can able to identify multiple antigenic determinants or multiple epitopes of the surface which are present on the surface of the antigen. So actually, we want monoclonal antibodies to treat any cancerous conditions, or we can able to fix the venom, that is snake poison, with the help of monoclonal antibodies. Apart from this, we have tremendous applications with the help of monoclonal antibodies. Apart from this, another difference is also there. Actually, polyclonal antibodies are producing inside our body, that is in our serum, in response to the antigen. Polyclonal antibodies will be produced. When coming to the monoclonal antibodies, these are produced in vitro conditions. That is only with the help of hybridoma technology, we can able to produce these monoclonal antibodies only in laboratory conditions. So in vivo procedure involves introduction to the hybridoma cells into the peritoneal cavity of the animal. Then from uh, ascitic fluid antibodies are isolated. In vitro method involves culturing of the hybridoma cells in suitable culture media, and then antibodies are isolated and purified. Once a hybridoma colony is established, it will continuously grow in culture medium because of the immortal nature, like RPMI 1640, and produce the number of antibodies. So these monoclonal antibodies can be stored in liquid nitrogen. So this is the pictorial diagram that is we can understand by looking at this representation, the production of monoclonal antibodies. So first of all, we are injecting the antigen to this mouse intraperitoneally, or intravenous injection we are giving, so that after 72 hours of the injection, we are getting the splenic B cells. These are having actually HGPRT enzyme. Apart from this, we are taking the myeloma cells which are lacking HGPRT. These two types of cells are fusing in the presence of polyethylene glycol. So we are getting a different types of cells in which we have to isolate only hybrid cells. That is possible with the help of hat medium. So we are getting the single cell of hybrid, single hybrid cell we are taking, and that is we are placing into the one well, that is one cell in each well, so that we are getting the clone number of copies of cells that are very much resembles to each other, that can able to detect only one single type of epitope of antigen. So we can treat the condition. Then we have to provide the suitable environmental conditions. These cells are multiplying into n number of cells. So after certain time period, these cells will produce the number of antibodies, that is monoclonal antibodies, which we want. That the monoclonal antibodies will be scrutinized by radioimmunoassay or enzyme-linked immunosorbent assay, so that we can store, after scrutinization, we can store the monoclonal antibodies with the help of liquid nitrogen. Next, coming to the applications of hybridoma technology, serological applications, that is identification of blood grouping and diagnosis, detection of pregnancy by assaying of hormones with monoclonal antibodies, and separation of one substance from a mixture of very similar molecules. Immunopurification, purification of individual interferons using monoclonal antibodies, and inactivation of T lymphocytes responsible for the resection of organ transplant. So some sort of immunosuppression also we can cause with the help of monoclonal antibodies. Not only serological tests, that is diagnosis, or serological typing, blood grouping. Apart from this, immunosuppression we can induce with the help of monoclonal antibodies. Then we can use these monoclonal antibodies for the treatment of cancerous conditions, various cancerous conditions, that is breast cancer, colorectal cancer, etc. Removal of tumor cell from the bone marrow, treatment of acute renal failure, treatment of malignant leukemic cells and B-cell lymphomas, etc. Apart from this, we can treat the arthritis, that is rheumatoid arthritis. We can treat the asthma. So number of applications are there for monoclonal antibodies. So that is why this topic is very, very important. So monoclonal antibodies we can produce only with the help of hybridoma technology. Thank you so much.